I'm really excited to present my thesis work entitled Bacterial Chemoprotective Mechanisms Rescue Post Anticancer Drug Toxicity. And if you don't know what some or all of those words mean, don't worry, we're going to go through it slowly. I'm going to start with an introduction, followed by 3 stories that make up the bulk of my PhD, and then a conclusion and acknowledgements. But before starting with the introduction, I'd like to pose a question. And that question is, what does it mean to be a human being? For millennia, our ancestors have asked what separates us from animals. They have come up with answers like love, sacrifice, cooperation, tool use. Yet, the more we learn of the natural world, the harder it is to argue that a honeybee does not cooperate or a crow does not use tools. In 1950, the physicist Alan Turing posed a slightly different answer to this question, asking what separates a human from a thinking machine. He proposed that you present the same question to a human and to a computer. If you can't tell the difference in the answers, we say the computer is capable of thought. With the advent of the transformer and large language models, modern computers can pass the Turing test with ease, further complicating the notion of what it means to be human. In 1952, the biochemist Rosalind Franklin collected data leading to the discovery of the structure of DNA. Finally, molecular biologists had a firm answer to the question of what it means to be human. After all, you and I share more than 99% of our DNA, more than with any other animal or any other organism. Yet, what about when our DNA betrays us, growing into cancer? What about when our DNA is not really ours, like when an HIV virus integrates itself into our genome? I don't claim to have an answer to this question, but I think the answer is incredibly important when thinking in particular about cancer care. And so I hope over the next hour, you can all think with me. We contain multitudes. Though we are but one species, Homo sapiens, inside each of our genomes is more than 20,000 genes, which together carry out the functions required for life. However, even though we are species, we contain more species inside of us. Inside every human's intestinal tract are trillions of gut bacteria. Recent estimates put the number at between 1 and 10 bacterial cells for every human cell. This is tremendously important for our biochemistry. These hundreds of species collectively encode hundreds of thousands of unique genes that complement and extend the biochemical potential of the human genome. Now, I'd like to present a clinical problem, the clinical problem of cancer care. Dose-limiting side effects pose a major barrier to cancer treatment. Cancer is fundamentally a disease of unregulated cell cycle division. Every time a cancer cell divides, it needs to make new DNA. This creates a therapeutic vulnerability. Perhaps we can target DNA synthesis. Many anti-cancer drugs work in exactly this way, targeting pathways involved in making DNA and rapidly killing cancer cells. Unfortunately, in our bodies, many of our own cells also rapidly divide, especially in the intestinal tract or in the hair follicles, leading to severe on-target but off-tumor toxicities that can lead to severe side effects. For my PhD, I studied one particular cancer drug, capecitabine. Capecitabine is a very widely used chemotherapy used to treat both breast and colon cancer. Unfortunately, capecitabine has high and variable toxicity. Up to 35% of patients on this medication require dose adjustments, and more than 10% of these patients require discontinuation from this drug entirely. This is due to adverse events like hand-foot syndrome or gastrointestinal toxicity. Think diarrhea. When I think about a drug that is highly variable between individuals, and a drug that is delivered orally and passes directly past all of our gut microbes, and a drug that targets a cancer that itself is in the intestinal tract, I wonder, can variation in the gut microbiome explain variation in the side effects of chemotherapy? I'd like to present my first story, a story of tiny pharmacists inside all of us. But to understand this story, I first need to introduce a little bit more biochemistry. Bear with me. Capecitabine, or CAP, is an oral chemotherapy that is sequentially activated and then biodegraded. It is given as a pill, a prodrug that is biochemically inert, activated by carboxyesterases in the liver and thymidine phosphorylase in the tumor into 5-fluorouracil, an active compound that inhibits DNA synthesis by blocking enzyme thymidylate synthase and disrupts RNA processing, killing cells, both cancer and rapidly dividing healthy cells. Inside each of our cells, we contain a gene called dihydropyrimidine dehydrogenase, or DPYD, which degrades active 5-fluorouracil into inactive dihydrofluorouracil. To make sure that we all understand what is going on here, I'd like to pose a thought experiment. What if there's a mutation in this degradation gene? Well, the downstream product will no longer be produced, and the active compound will accumulate, leading to more cell killing, both of tumor and of healthy cells. This is not just an academic exercise. In 2022, David McIntyre passed away by having a mutation in this exact gene and being dosed with this drug. This then begs the question, why study the gut microbiome? After all, I've just told you that there's a human gene that matters a lot for this conversion. Inside of our bacteria, there is a gene that is homologous in structure and function to human drug clearance enzyme DPYD. This bacterial operon and protein are called PreTA. And with this in mind, I'd like to introduce you to our clinical study, the gut microbiome and oral fluoropermitine clinical study. Designed and headed by Wes Kidder, sitting back there, this study was designed in 2018 and intended to assess the role of the microbiome during cancer treatment. This study enrolled 40 patients with colorectal cancer to one of 3 different treatment groups, all receiving oral fluoroperimidines, chemotherapies that have biochemistry in common with capecitabine. During this study, patients took chemotherapy for 2 weeks daily, followed by 1 recovery week, as is common in standard of care. During this study, these patients generously donated and collected their stool, froze it in their home freezers, shipped it to us, both before, during, and after treatment, enabling us to ask longitudinal questions of what is happening to the microbiome during cancer care. To assess the microbiome, I'd like to introduce a technology that some of you may have used before and some of you may have not. And that technology involves DNA sequencing. We collect the stool, which contains all of the microorganisms that pass through the intestines and out into the stool. We then extract DNA from the stool. In the context of the GO study, the vast majority of this DNA extraction had already been done for me before I joined the lab by former MD PhD student Than Kya. We then infer which bacteria contributed different strands of DNA using various databases and computational methods that I developed in tandem with Cecilia Naker. This lets us ask our first questions related to actual data, like how does the chemotherapy impact gut microbiome diversity in patients? Well, this seems a simple question. I'd like to just highlight, it looks like between the top graph and the bottom graph, there are fewer bacteria post-treatment. And in fact, this is what we saw in our study. Using a metric called Shannon entropy or Shannon diversity, we observed a decline in diversity within days of initiating treatment. Seeing that there's a change in diversity made me wonder which bacteria are most impacted by chemotherapy. And so I asked the question, are there certain bacteria, say the orange ones that are suppressed by chemotherapy, or others, say the blue ones that are enriched by chemotherapy? After performing some linear mixed modeling, I determined that there are 27 bacteria that are significantly decreased during treatment and 16 that are significantly increased during treatment. Feeling fairly convinced that something is happening to the microbiome during cancer treatment, I wondered, why does the microbiome change during chemotherapy? Our hypothesis as a lab had always been the drug is directly killing certain bacteria. But many of these patients are receiving other drugs or concurrent therapies, which could also impact the bacteria. And many of these patients have cancer and lots of other complicated stuff going on, which could mediate the effect of drug on the microbiome. To address this question, I first asked, does capecitabine impact the microbiome in the same way across all 3 of our subclinical cohorts, or clinical subcohorts? I observed that within the group treated with capecitabine only, within the group treated with TAS-102, another oral fluoroperimidine, and within the group treated with immunotherapy plus capecitabine, when the signal appears, when there are enough bacteria to be detected that are of the same type, the signal is largely consistent. And a power analysis that I won't show here showed that we expect the lack of signal in the latter two cohorts is due to the small sample size. Next, I wonder, can we assess this problem in a more controlled experimental fashion? Specifically, can capecitabine change the microbiome in a mouse model? To do this, I collected stool from mice, and then I treated them with either chemotherapy or vehicle for 4 days, followed by another stool collection. This type of plot is called a principal coordinate analysis, and it embeds the high-dimensional microbiome composition in a 2-dimensional map that we can all look at. When looking at mice that were treated or went on to be treated with either vehicle, which is just what we call a control group in biology, or capecitabine, chemotherapy, we found that they're not super different at baseline. And if we look at the day 4 vehicle, it's also not super different than baseline. However, in the mice that were treated with chemotherapy, their microbiota composition differs dramatically from their baseline microbiota composition, suggesting that indeed capecitabine impacts the microbiome in mice. I also wondered, does capecitabine impact the microbiome in a test tube in vitro? To do this, I took stool from 3 patients and incubated the stool with either capecitabine, its active compound, 5-fluorouracil, or a vehicle control, and then performed sequencing on the ex vivo communities to see how the composition may have shifted. From one of the donors, we can see that at baseline, post-treatment, the vehicle points cluster together, but the communities that were treated with 5-fluorouracil or capecitabine differ substantially from the vehicle, suggesting again that capecitabine impacts gut microbiome composition in vitro. When we compare these results to the other 2 donors, we see that consistently there is a difference between the vehicle and the chemotherapy-treated communities. However, this difference is very inconsistent across different donors, suggesting that there is likely high inter-individual variability in how the microbiome responds to treatment. Now that I saw the microbiome changes during treatment, I wondered, which parts of the microbiome are changing? How does chemotherapy impact the specific genes in different microbiota? For this, we used a technique called metagenomic sequencing to assess if there are certain genes that go down, the orange genes, or genes that go up, the blue genes. After performing another linear mixed effects model, I observed that hundreds of gene families were increased following treatment, leading me to wonder, what do all of these genes have in common? Performing a gene set enrichment analysis, I found that pyrimidine metabolism genes were consistently enriched following treatment in the patients. Now, why does this matter? To bring you back to the background, capecitabine is converted to 5-FU, which is then inactivated by pre-TA. Pre-TA is a pyrimidine metabolism gene, and it is one of the genes that drove that signal in that last graph. In particular, if we look at pre-TA abundance over time, within a few days of capecitabine initiation, it has increased slightly, and within many days of capecitabine initiation, it has increased by many-fold. This makes me think capecitabine chemotherapy alters the gut microbiome directly, enriching for bacterial drug clearance gene pre-TA. This makes me wonder, how does pre-TA impact cancer treatment? To do this, I had to develop a model of mouse capecitabine toxicity because there was no model in the lab nor in the literature. I treated mice for 4 days or 12 days with chemotherapy and assessed the following endpoints, including weight loss as a metric of acute toxicity and colon length as a metric of intestinal symptoms. Many other endpoints were assessed, but will not be discussed today. My first question was, in this group of either vehicle or chemotherapy treated mice, how does chemotherapy impact the body weight and the body composition of these mice? Over the time of treatment, the mice treated with capecitabine lost up to 5% of their body weight, with one mouse even losing nearly 15% of its body weight, whereas mice treated with vehicle remain at a stable weight as expected. We also wonder, is this because of differences in fat percentage or lean percentage? Or anything else, and we observed that there's no difference in body composition of these mice, suggesting this is really an acute loss of all types of tissue, both fat and lean tissue. Next, we wondered, how does capecitabine impact gastrointestinal toxicity, a correlate of those really common clinical symptoms of diarrhea? In mice, interestingly, when the mouse colon is subjected to damage, the colon literally becomes anatomically shorter, and you can measure this difference. Within 4 days of capecitabine treatment, the colons of the capecitabine treated mice were shorter than the colons of the vehicle treated mice. And the same thing was true at day 12. We also measured a molecular correlate of intestinal inflammation called lipocalin-2, observing not much of an increase by day 4, but a significant increase by day 12, making us somewhat confident that our mouse model recapitulates both acute toxicity and gastrointestinal phenotypes. Next, I wondered, does the microbiome matter at all? How does presence of a microbiome matter in this mouse toxicity model? To do this, I'd like to introduce 3 terms that are common to us in the microbiome field, but perhaps not common to those outside. One is a conventionally raised animal. This is a normal mouse with its normal bacteria living in a normal facility. Another is a germ-free mouse. This is a special mouse who is raised with no microbes, living in a special little isolator that you reach into with gloves to work with. And finally, a conventionalized mouse. This is a mouse that eats the poop of a conventionally raised mouse to transition from being germ-free into having a microbiome once again. When I dosed all of these mice with capecitabine chemotherapy, I observed that over time, the mice that were germ-free lost a considerable amount of weight, while the mice that were either conventionally raised or conventionally derived, in other words, mice with a microbiome, did not experience nearly as severe toxicity. I performed a follow-up experiment wondering, can we recapitulate this effect with antibiotic treatment? I treated mice with either a vehicle or broad-spectrum antibiotic, and then began dosing them with chemotherapy. I observed that this time within even 5 days of treatment, the antibiotic-treated mice lost large amounts of weight, suggesting once again that absence of a microbiome leads to more severe chemotherapy toxicity. Finally, I wondered, how does pre-TA play into this story? I showed that the microbiome matters for toxicity, but does this one gene matter at all? To do this experiment, I worked with the postdoc Taylor in our lab, and we depleted the microbiota of mice using streptomycin. We then colonized them with these engineered strains of bacteria, E. coli that contains no pre-TA gene, E. coli that cannot metabolize drug, versus E. coli that contains extra expression of the pre-TA gene, E. coli that's a super metabolizer of drug, followed by treatment with chemotherapy. Once again, I tracked the weight over time, and I observed a pretty striking difference between the groups. In the mice that are treated with bacteria that can clear active drug in their intestines, these mice did not lose body weight over 8 days. In mice treated with wild-type E. coli, which is an intermediate drug metabolizer, these mice lost a little bit of weight. And in mice treated with E. coli that has no drug metabolism capacity, these mice lost a lot of weight. The same thing was true across the colon length phenotype. Next, I wondered, why is pre-TA impacting side effects? We have a strong hypothesis that pre-TA is directly metabolizing 5-fluorouracil. So we had to measure 5-fluorouracil. And to do this, we once again use these streptomycin-resistant E. coli that are identical except for pre-TA. So just to recap, no pre-TA, no drug clearance. Extra pre-TA means extra drug clearance. Once again, I depleted the microbiota of mice using streptomycin, followed by giving the mice engineered bacteria that either do or don't have the capacity for this particular drug metabolism, followed by an oral pharmacokinetics experiment. What this means is I gave the mouse a drug, and then I collected stool for 7 or 8 hours regularly to see how much drug is in the stool after certain amounts of time. The first drug we wanted to measure in the stool was capecitabine. After extensive comments from a reviewer wondering if capecitabine even is in the stool, and we observed that in both groups, there were similar amounts of capecitabine detected in the stool over time. In other words, when you give a mouse a drug orally, the drug does indeed, some of it makes it all the way to the stool and is measurable. And this amount of inactive drug is similar between groups colonized with different bacteria. However, when we measured levels of active drug of 5-fluorouracil, we saw that we could also detect 5-fluorouracil in the stool. And once again, there's a slight decline over time as the mouse and its bacteria clear this 5-fluorouracil. However, in mice that in their intestinal tracts have the capacity to clear all of this drug, these mice have substantially lower levels of 5-fluorouracil in their intestines, potentially explaining why there's a difference in toxicity between one mouse colonized with super metabolizing bacteria and a mouse colonized with a non-metabolizing bacteria. Finally, I sought to connect all of this back to the patients that enrolled in our trial, wondering, do patients with side effects have different levels of pre-TA, this drug metabolism gene? In our trial, inside of the chart, 21 out of 40 patients documented at least some type of toxicity, with most toxicities being hand-foot syndrome or gastrointestinal toxicities. When we compared the group of patients who documented no toxicity versus those who reported toxicity severe enough that made it into the chart, we saw a significant difference in pre-TA levels, suggesting that in patients, potentially pre-TA is playing some role in the development of chemotherapy side effects. We then wondered, can we go a step further? Can we use pre-TA to actually predict side effects in patients? And to do this, we use something called a receiver operating characteristic curve. And to give you a context, if you haven't seen this kind of curve before, if you have a square line like this is a perfect prediction. We say the area under the curve, or AUC, is 1. If you have a diagonal line, this is a terrible prediction. This is no better than random chance, and it has an AUC of 0.5. When we look at our data, we have an AUC of 0.68. This is Not good, but not bad. It suggests that the microbiome likely plays some significant biological role, but also that it's not the whole story. But we knew that already. We know that things like host genetics, things like diet, things like sex matter for drug metabolism. And so perhaps more than anything, the microbiome is just one additional tool in the pharmacokinetic toolkit. To see how pre-TA compares to all other microbial genes, pre-TA is one of the best predictors of all of the tens of thousands of microbial genes that were tested, suggesting that this is a true biological signal. Finally, I wondered, we were looking at pre-TA versus any documented toxicity. This can include things as severe as dose-limiting diarrhea, or things as common as nausea or fatigue, which are almost ubiquitous among patients taking chemotherapy. So I instead decided to say, okay, what if we look at just patients who had to have their dose adjusted because the toxicities were so severe? And pre-TA actually performs a little better in these patients, suggesting that potentially patients who have very high bacterial drug metabolism are less likely to experience really clinically significant dose adjustments. To summarize, bacterial drug clearance gene, pre-TA, can predict and prevent side effects in mice and maybe predict side effects in patients. In summary of the tiny pharmacist story, using mouse models, human studies, and in vitro experiments, and with our background knowledge that the gut microbiome operon pre-TA can clear active chemotherapy, we discovered that chemotherapy selects for bacteria containing pre-TA. It increases the levels of pre-TA post-treatment. This bacterial pre-TA can prevent side effects in mice, and bacterial pre-TA can maybe predict side effects in patients. I've told you a story of the pharmacists that live inside of us, and now I'd like to tell you a story of how you can control them. Your microbiome is what you eat. My advisor, Peter, has done much fundamental work studying how diet alters the microbiome. But one of my favorite studies that he's involved in enrolled 10 subjects. These subjects were asked to eat their normal diet, and their microbial diversity was tracked over time. These subjects were then asked to switch to eating an entirely animal-based diet, and their microbiome diverged dramatically from baseline within a period of days. When these subjects were asked to return to eating their normal diet, their microbiome diverged back towards their normal state, suggesting that diet can alter the microbiome in a very controllable way and within days. I'd also like to provide an additional framing background question, which is why should we think about diet in the context of cancer? This is because extensive epidemiological literature suggests that diet impacts colorectal cancer risk. I can see that many of you are getting sleepy. I also know that many of you know the correct answer to this question. So I'd like to conduct a small poll. For each item I list, I want you to put your thumbs up if you think it's good, it prevents cancer, and thumbs down if you think it's bad and causes cancer. So let's start with alcohol. Now, most people have thumbs down. That's correct. Alcohol is associated with increased colorectal cancer risk. How about red meat? Also, a lot of thumbs down. You guys all hate meat. Yeah, red meat is also associated with increased colorectal cancer risk. How about dairy? You're like some thumbs up, some thumbs down. I was a little surprised by this one. Lots of epidemiological studies show that dairy may be protective against colorectal cancer risk. And what about fiber? Our favorite thing to study is gut microbiome. Everyone put their thumbs up. There is only weak evidence that fiber is actually beneficial. This is just to provide a framing question that epidemiologically, we know diet matters. And for this reason, any changes in diet during chemotherapy are potentially clinically significant. Then we ask, how does diet change during chemotherapy? And I've told you about the design of the GO study, but what I didn't mention is we had a fantastic epidemiologist and dietitian working with us, Erin Van Blerigen, who said, you guys should really conduct diet surveys. And fortunately, diet surveys were included as part of this GO clinical study, which meant for the 3 days before each of their doses, these patients spent inordinate amounts of time filling out really detailed paperwork of everything they ate. This is over a 72-hour period every time you see an orange dot. It's really very onerous. I filled them out myself to see how hard it was. And I'm very grateful that the patients took the time out of their cancer treatment to do this study for us. Having the dietary survey information in hand lets us ask several questions, including the question of how does diet quality change during chemotherapy? To talk about diet quality, I'd like to introduce something called the Healthy Eating Index, or HEI. This is a metric that broadly captures diet quality. It ranges from 0 to 100. It's associated with health outcomes ranging from cancer to cardiovascular disease. And the components are things that you might expect. things like saturated fats and trans fats, things like refined grains versus whole grains, things like eating enough fruits and vegetables every day, things like not eating too many added sugars. These patients at baseline before treatment averaged a healthy eating index of 60 out of 100, suggesting something we already know, which is one, Americans on average have a healthy eating index around this range. And two, it's also possible that the cancer itself makes it hard to eat a more complete diet. This is a cancer in the intestinal tract that causes intestinal symptoms. Within initiation of their first cycle, the healthy eating index had already decreased. And by cycle 3, it was significantly lower than at cycle 1, suggesting that over the course of chemotherapy, diet quality changes. You then wonder which components of the healthy eating index are shaping this decrease over time. And after investigating the 13 components, only 2 were decreased over time: the refined grains score and the fatty acid score. What this means is that these patients over time are eating more refined grains and fewer healthy fatty acids. If we remove these 2 scores from the calculation of the healthy eating index, there is no difference over time. In other words, the change in refined grains and the change in fatty acids is entirely driving this decrease in diet quality during chemotherapy. Now that we've looked at diet quality broadly, I turned my attention to specific nutrients and food groups, asking how do nutrients and food groups change during chemotherapy? To do this, I assessed the 79 dietary variables that were measured by the ASA 24 survey, finding that refined grains were enriched following treatment, and a depletion in nuts and seeds, as well as several fat-soluble micronutrients after treatment. I'd like to show a few exemplary graphs just to show that these are indeed going down, and this isn't some statistical trick in the left-hand plot. Vitamin K1, which comes from dark leafy greens, was decreased following chemotherapy. Nuts and seeds intake was also decreased, and refined grains intake was increased following chemotherapy. Next, I wondered, I've just told you that microbiome changes during chemotherapy. Do these changes in diet explain changes in the microbiome during chemotherapy? To ask this question, I used the 43 bacteria that I previously identified as changing with chemotherapy. And I actually filtered them to an even smaller list. I required bacteria be present in at least 10% of samples, and I required a sufficient amount of paired data for analysis, leaving me with 15 bacteria where we could robustly probe diet bacteria interactions. I then performed a relatively simple analysis correlating the change in bacteria with a change in each diet variable between any 2 consecutive within patient time points. And we observed that there were only 2 really significant positive interactions. A positive interaction between lactobacillus and vitamin B12, which we later found out is because vitamin B12 comes from yogurt and lactobacillus also comes from yogurt. And so these patients are eating vitamin B12 and lactobacillus at the same time. But we also discovered this other, maybe more surprising interaction between a species called Faecalibacterium prausnitzii and vitamin K1. Why did I care that I saw this bacteria? Well, Faecalibacterium prausnitzii is really interesting. There have been extensive studies, including the one cited here, that demonstrate that Faecalibacterium prausnitzii can actually improve response to immunotherapies, both in mouse models and predict response in human patients, suggesting that understanding what happens to Faecalibacterium matters, especially when we think about adding immunotherapy onto traditional chemotherapy regimens. So how did vitamin K1 intake impact Faecalibacterium prausnitzii? I stratified patients into patients who either maintained or increased their vitamin K1 intake between cycle 1 and cycle 3, or patients who decreased their vitamin K1 intake. In patients who increased their vitamin K1 intake, we see a decrease in Faecalibacterium with time, a decrease by about a factor of 2 or 3. Note that y-axis is log scaled. However, in patients who decrease their vitamin K intake, we see a much larger decrease in Faecalibacterium prausnitzii, suggesting that Faecalibacterium prausnitzii positively interacts with vitamin K1 intake. Next, I wanted to zoom in on one other specific bacterium. This is a bacterium some of you may have heard of before, but many of you have not. A bacterium called Fusobacterium nucleatum. This is the most studied bacterium in colorectal cancer. It is enriched and found in nearly 100% of colorectal cancer patient gut microbiomes. It is enriched in the tumor next to the tissue, and it has been shown to directly cause cancer in APC-min mouse models. However, when I looked into my data sets, I saw that using 16S sequencing and metagenomic sequencing, I detected this in less than 25% of my samples, So I turned to a more sensitive method of detection, carrying out an assay called qPCR on all of my samples, where I found, more in line with my expectations, Faecalibacterium was present in nearly every patient sample. I then wondered, okay, Fusobacterium. How does Fusobacterium change during chemotherapy? I actually had a really strong hypothesis about this. I did a journal club at the beginning of grad school, which did a huge drug screen and showed that the chemotherapy 5-FU inhibits Fusobacterium nucleatum. That is the chemotherapy that we are more or less studying. However, when I looked at Fusobacterium abundance versus time, we have two expectations. If it decreases, then the capecitabine inhibits Fusobacterium. But if it's flat, we might conclude capecitabine does not inhibit Fusobacterium. And I observed that this was flat. In our data set, there was no change in Fusobacterium abundance, maybe a slight dip at day 3 over time. But then I wondered, is this effect maybe mediated by something else? In other words, do any of the dietary micronutrients impact Fusobacterium abundance in patients? Performing an analysis of the interaction between Fusobacterium and every dietary nutrient, I found very little signal, but one significant signal, a negative interaction between copper and Fusobacterium. I wondered, where is this copper coming from? I don't really know where copper comes from in diets. I'm not eating a metal bar. And so I looked back in our data, and the copper is coming a lot from nuts and seeds, which we know are changing with treatment, a bit from grains and starches. We know those are increasing with treatment, so it's a complicated story, and a lot coming from other stuff too. So if you're worried about copper deficiency, don't be. I then wondered, how is this a real signal, or is this just a statistical chance that we observed in our big human study? And so I asked the question, how do chemotherapy and copper impact Fusobacterium in a test tube? I took Fusobacterium and treated it with or without chemotherapy in the presence or absence of copper. When the Fusobacterium is treated just as a control, it grows to 100% of its potential. When it is treated with copper alone, there is a slight suppression, but when it's treated with 5-fluorouracil, there's a large decrease in Fusobacterium. This is in line with the level of results and the sensitivity reported of Fusobacterium to 5-FU earlier. However, when we treat with both 5-fluorouracil and copper, we observe almost no growth of Fusobacterium, a synergistic effect consistent with the signal that we observed in patients. In summary, using both human studies and in vitro follow-on experiments, and knowing that gut bacteria matter for cancer development and treatment, we found that dietary intake changes during chemotherapy, with a decrease in nuts and seeds and a decrease in plant intake, as well as an increase in refined grain intake. We found that this decreased vitamin K1 intake can explain decreased Faecalibacterium prausnitzii. And we found that copper suppresses Fusobacterium, definitely in vitro and potentially in patients. I'd like to tell one final story. And on the dietary theme, this one is called Micronutrients, Macro Effects. The key onus of this story was, do any of these findings generalize outside of the 40 patients at UCSF who happened to enroll in our clinical study? Early on in my PhD, a group led by Marjolein Smit in the Netherlands published a paper that we thought completely scooped us. After reading the paper, we realized that everything was fine, and we set up a call with them. And they said, we've got all of these samples. We have all this poop in the freezer. Do you guys want it? And thus began one of the most exciting international collaborations of my PhD. This study enrolled 56 patients with colorectal cancer in the Netherlands. These patients were treated with capecitabine for 2 weeks, followed by 1-week recovery periods for 3 cycles. This clinical study collected stool at baseline before treatment, in the middle of cycle 3, and post-treatment. Slightly different time points than our study, but at least it's somewhat similar in design. The first question I had after I received the stool, performed DNA extraction, metagenomic sequencing, metadata cleaning, etc., was, do our pre-TA results, our results about drug metabolism, hold in this cohort? And I was delighted to find that, similar to in our own cohort at UCSF, we observed an increase with pre-TA over the course of chemotherapy, and we observed a strong relationship between pre-TA abundance and dose adjustments, leading me to believe that my pre-TA story is robust. But I have this really rich data set. We've set up this whole collaboration. We waited 1.5 years for the material transfer agreement to come through. What else can I learn from this cohort that we worked so hard to get? And one of the things that the clinical team in the Netherlands collected was really incredible prior exposure data. They collected things like prior antibiotics, prior systemic treatment, tumor sidedness, sex, prior proton pump inhibitor use, which people think impacts different parts of the microbiome. And so I compared all of these clinical variables with the baseline microbiome across patients. And I found the two variables that had the strongest effect on the microbiome was prior antibiotic use and prior systemic treatment. When I say prior systemic treatment, I mean prior chemotherapy, and usually prior chemotherapy with 5-fluorouracil-based regimens, or capecitabine. To tease apart this effect a little further, if we look at patients who received neither antibiotics or systemic treatment before enrolling in this trial, they have a Shannon diversity index of about 4 or slightly higher. If we look at patients who received antibiotics alone, they have a statistically significant, but maybe not a huge effect size, less diversity. Interestingly, if we look at patients who received chemotherapy prior to enrolling the study, we see a similar decrease in diversity. And finally, when we look at patients who received both antibiotics and systemic treatment, we see an even stronger decrease in diversity. This, although the graph might not be the largest effect size you can imagine, was incredibly interesting to us for two reasons. The first reason is that in this cohort, antibiotics and chemotherapy left a similar scar on the microbiome. This scar lasted for months after the initial treatment. An exclusion criteria for this study was these patients could not have been on antibiotics in the past several months, which means an exposure that they had months ago is impacting their diversity when we measured them. Second, it told us that it's important to think about the synergy of different drugs acting together, something that I never quite addressed in completion during my PhD. I then wondered, in this cohort, how does chemotherapy impact the gut microbiome gene pathways? And I observed, once again, hundreds of pathways that are far too many to manually go through are enriched following chemotherapy. But then when I opened the list, it actually became really easy. 7 of the top 10 just said menaquinone biosynthesis pathway. And so this made me wonder, this is really fascinating. 7 out of these top 10 hits are the same synthesis pathways. We're seeing some signal in patients. Let me go, dig up some other data. And the other data I dug up was from an experiment called a transposon sequencing experiment. In collaboration with Adam Deutschbauer, Benjamin Guthrie, a former postdoc in our lab, had conducted and then never analyzed incredibly complicated experiment where he took a library of bacteria. Every single one of these bacteria has a knockout in one specific gene. Then he treated these bacteria with either vehicle or with a drug, 5-fluorouracil or capecitabine. Now, it's a little complicated to think about what happens, and I think Peter and I have both messed up on presenting this graphic. But when you knock out a gene with a transposon, if the gene knockout is harmful, so green or blue don't show up in the drug treatment, that means the gene itself is good. The gene is protective. Conversely, if the gene knockout is beneficial, that means the gene is bad. The gene sensitizes the bacteria to the drug. It won't matter if you don't get this. So I'll just move on to the results, which is which genes protect bacteria in vitro. We observed that hundreds of genes protect bacteria from 5-FU and significantly fewer from different prodrugs of 5-FU. Importantly, I did a gene set enrichment analysis on every gene from all of these drug treatments, and there was only one significant gene set. This gene set was quinone biosynthesis. Now I had a signal from the human study and a signal from this in vitro experiment pointing towards exactly the same pathway. So now I had to do some reading and figure out what is menaquinone. And so I learned that menaquinone, referred to as MKN, where N denotes the side chain length, is more commonly referred to as vitamin K2. I put the structure up here for any chemists, mostly Chen. This vitamin is really fascinating because we cannot make it. Human cells do not have the capacity to make any type of menaquinone of any side chain length. This is a gene that is made by bacteria. As a result, when it comes into us through our diet, this is primarily through meat and through fermented foods. The graphic on the right is natto, which Ethan fed me for the first time in Japan this summer. I then wondered, okay, we have a signal that menaquinone synthesis is increasing after chemotherapy in patients. We have a signal that menaquinone is protective in vitro. What if I just look at menaquinone? Does vitamin K2 protect E. coli alone from chemotherapy toxicity? To do this experiment, I took E. coli, either wild type, the normal lab strain, or an E. coli that is unable to synthesize vitamin K2. And I treated these E. coli with 5-FU, the chemotherapy, and then with or without menaquinone, this vitamin K2. I observed that the wild type E. coli grew to an optical density of about 0.22. But under 5-FU pressure, the E. coli that cannot synthesize vitamin K2 grows significantly less. However, when we supplement the E. coli that cannot grow vitamin K2 with vitamin K2, they grow to nearly the same level as the wild type, suggesting that vitamin K2 protects E. coli from chemotherapy side effects. Now that I've thought about E. coli side effects, I'll get back to who I actually care about side effects for, which is patients. And so I wondered which patient side effects are most explained by the gut microbiome. To ask this question, I once again performed sort of a permutational analysis of variance, assessing which side effects explain most variability in the gut microbiome. And although there were 3 significant results, the most significant result was a relationship between peripheral sensory neuropathy, think really painful tingling of the hands and feet, cold sensitivity, and the microbiome. Now that I knew there's theoretically some relationship, I decided to dive deeper and asked which pathways explain this peripheral neuropathy. I observed this interesting sort of asymmetrical volcano plot. All of the orange pathways on the left are lower in patients who go on to develop peripheral sensory neuropathy. To reiterate, this is comparing the baseline levels of microbiome pathways to future development of a side effect on chemotherapy. But when I looked at my top 10 hits again, 3 out of 10 of those hits were menaquinol biosynthesis pathways. Now, using 3 unbiased analyses from 3 different directions, we had identified menaquinol as potentially clinically significant. I then looked at the underlying menaquinol data, and not surprisingly, given that it showed up as a significant hit, patients who have lower levels of baseline vitamin K2 from their microbiome also experience lower levels of peripheral sensory neuropathy during treatment. We went on to validate that the gene levels also correspond to the actual vitamin K2 levels with the metabolite measurement in collaboration with Tufts. I then wondered, does this matter in mice? And to study this in mice, I first had to figure out how I study neuropathy in mice. And I did the best I could, which is finding a study published on capecitabine toxicity in 2023, which used a test. Put a mouse on a 52 degrees Celsius hot plate. This is kind of hot. If you put your hand on something 52 degrees, you will withdraw it after some amount of time. If your hand is more sensitive, you will withdraw it faster. The test is very simple. You measure the time until the mouse flicks its paw or jumps or licks its paw, trying to withdraw from the hot stimulus. With hot plate test in hand, I performed an experiment where I tested the sensitivity of mice at baseline and at day 12 following capecitabine treatment. This was just to establish the model. There is no vitamin K2 in this experiment yet. I observed at baseline the paw withdrawal latency of the mice was similar between groups. By day 4, there's maybe a little bit of a difference. And by day 12, there was a significant difference in paw withdrawal latency between the different mice, leading me to conclude that I had a way to measure paw sensitivity, a correlate of peripheral neuropathy in a capecitabine toxicity mouse model. Next, I wondered, how does vitamin K2 impact this symptom in mice? To do this, I enrolled 32 mice, treated them with either vehicle or vitamin K2, and I treated all of the mice with chemotherapy. I collected body weights every day. I collected colon length at the experimental endpoint, and I performed a hot plate test both before and after chemotherapy. I observed that the mice in both groups lost pretty similar amounts of weight to each other. The mice in both groups had a similar colon length after capecitabine treatment. But in the vehicle-treated mice become much more sensitive. Their paws are much more sensitive after treatment. They withdraw their paws 5 seconds sooner. However, in the vitamin K2 treated mice, on average, these mice do not withdraw their paws any sooner than before baseline. In other words, vitamin K2 rescues this peripheral neuropathy symptom, but only this peripheral neuropathy symptom in mice. In summary, using human experiments, mouse models, and in vitro studies, we determined that chemotherapy selects for bacterial vitamin K2 genes, that vitamin K2 is associated with side effects, in particular peripheral sensory neuropathy in patients, and that vitamin K2 can prevent peripheral neuropathy-like symptoms in mice. I'd now like to offer a brief conclusion and prospectus. Our gut bacteria transform our drugs. Pre-TA metabolizes 5-FU to dihydrofluorouracil, impacting side effects. Our gut bacteria are shaped by our diet. Decisions that we make every day impact the microbes that are our passengers. And our gut bacteria produce micronutrients that we cannot, micronutrients that are potentially clinically relevant. In cancer treatment, I think that instead of considering human beings as organisms, we must consider the full complexity of the meta-organisms on and inside of us. In terms of ongoing work related to this project, postdoc Taylor in our lab is working on studying how defects in host drug metabolism impact some of these phenotypes. A postdoc Edwin is working on how circadian rhythms shape some of these interactions. And I'm continuing to work with Evan Walker in GI cancer to study diet-drug microbiome interactions in the context of pancreatic cancer treatment. I'd like to pause for a moment for some questions. Thank you.